Okay, so things to remember. We're going to work on those models, finishing up the DNA replication models we started on yesterday. And there's a protein synthesis activity for those of you at home that should be linked for you in there. And then I wanted to mention this. On Wednesday, day 2, you guys had data set 1, is that correct? You were supposed to do data set 1 on day 2? Good. We were supposed to do data set 2 on day 3. So we didn't have time for that yesterday, and we're not going to have time for it today. So by Monday, make sure that you have went online in days 2 and 3 and completed data sets 1 and 2. It shouldn't take more than a couple minutes. Okay, the pictures should be in there that go along with them. Go in, answer whatever you think it is. And those are only about 5 points each. But hopefully y'all are getting better at those now, looking at the graphics, using the information and knowledge that you have from our discussions to answer those. Whew. All right, what else? Today we're going over trying to finish up protein synthesis part 1 and make sense of it. And then if we get a chance, we may start a little bit of part 2, but I don't know if we'll get there because this is not easy by any means. I would say these two concepts, replication and protein synthesis, are definitely two of the most important ones you need to be familiar with. So if you guys will turn to 338, and yesterday we left off at codons, is that correct? Yes, that is correct. All right, codons, what does that sound a lot like? Code. Okay, what do codons code for? You're close. What makes up proteins? What are the monomers of proteins? Amino acids, good. So codons code for amino acids. Codons are determined by the template strand of DNA, the blueprint information. So remember, DNA is your main blueprint, and then little cheap copies are made from that, and then they're recycled, and then make some more if you need them. One other important thing to remember, DNA and RNA is always copied and always read in which direction? 5 to 3. So make sure that you always do that. B, the codon chart. So before we get to the codon chart, let's look on 338 here. You've got a sample strand of DNA, and actually let's back up to 337 real quick. Just make sure we're all on the same page. Remember, at the top of the page, prokaryotes. You have a segment of DNA that's copied into mRNA. What do we call that segment of DNA? What is a segment of DNA that's going to code for a protein that's copied into mRNA called? A gene, good. So a gene is a segment of DNA that's going to code for a specific protein. A couple other things to remember. You know, a lot of those proteins, they're either going to make up something in our body, 50% of our dry mass is protein, or it's going to be used to produce some type of enzyme that will be help facilitate some type of reaction that's taking place in our body. Remember, almost all the reactions in our body are facilitated by enzymes made from proteins. Now, there are enzymes made from, you might remember the other one? RNA, ribozymes. Talked about that earlier in the year. But most of them are going to be constructed from protein, okay? So, on the bottom of 337, remember, there's a little bit of difficulty in eukaryotic cells, though. Eukaryotic cells, you don't just copy the DNA and it goes out and makes a protein. It has to be modified through RNA processing, where you've got to remove some stuff and you got to stitch some stuff back together. So it's a little bit more time consuming, but we are going to get into that toward the end of the notes today. Okay. So on 338, we've got a strand of DNA at the top here. Now, DNA has 2 sides. You're only going to copy one side. One side's called the sense strand. One side's called the nonsense strand. I would venture to say they're going to tell you, or I'm going to tell you which side to copy. So don't freak out. I don't know. To be honest, I don't even know by just looking at one how they figure out which one's getting copied and which one's not. So you will be told which side gets copied. Now, on the case of 338, the template strand that's copied, which direction is it running from left to right? My eyes red on the outside right there, Martin. Then you take a look at that. All right, the template strand, which direction is it running from left to right? The DNA template strand on 338. 3 to 5. So if it's going 3 to 5, then the RNA that's copied that matches up with it is going to go which direction? 5 to 3. Good. So remember, just like with the DNA, when the mRNA is copied onto that template strand, it's going to be running anti-parallel. Okay? So, thanks. So it's going to be copied. Now, if you look, what are the first 3 nucleotides on the DNA that are copied? ACC. So A goes with, C goes with, C goes with. So what you're going to be tasked with a little bit later, and we're going to do in the lab next week, is you're going to have a strand of DNA, and then you're going to have to figure out what mRNA would go with it, and then you're going to have to use that mRNA in order to code out the protein, okay? And you can see below it here, UGG. Now, mRNA, what do we call those first 3 letters? Or each 3-letter segment? A codon. So those 3-letter segments code for a specific amino acid. Now, you will be given the chart or a chart similar to this on 339 on the test. And they will say, here's your DNA template strand, code out which protein would go with this. And you would have to figure out the protein, okay? Now, you'll notice, how many amino acids did we say were in there? 20. How many different codes do we have? Mariah, you know this.
64.
64. Why do we have 64 different codes for 20 different amino acids? That's a good question. There are many multiples. If you look here in the codon chart, some of them like leucine, the LEU, or serine, or pro, whatever that is, or they got the abbreviations here. You don't need to know them all because obviously I don't. Many of them, there are multiple different codes that go with them. And you could see like on the serine, the SER, what happens even if you have a mutation in that last letter of the codon? It's still going to code for the exact same thing. That's exactly right. And that's true for many of these. So what those extra codons do is even if you have an alteration in one of the letters, it will still code for the same amino acid and it won't affect your protein. And we talked about how significant that is where in sickle cell, where you have one letter, one nucleotide that's off, and it can cause a massive distortion in the way that protein is supposed to fold, in the way that the functionality of the protein hemoglobin works out to be in people with sickle cell. So, we good so far? So, let's see on our notes. Codon chart contains 20 amino acids. Nuremberg figured this out, figured out the first one. The UUU, congratulations. 61 of the 64 possible codons codes for an amino acid. The other 3 do what? There are stop codons. So you can think, and I think the lab we do on Tuesday will be like this. You can analogize a protein is similar to the making of a protein is similar to making a sentence. At the beginning of a sentence, how do we know we have a beginning? Yeah, so the word's capitalized. So you see that at the beginning. So we also have a start codon that indicates where we start out at. The start codon is AUG. I don't think you need to memorize that. Typically that's listed on the table. But that's where what you're looking for where to start. Because remember, you got these long segments of DNA, And when enzymes go to attach to this DNA, they are looking for specific sequences to know where to attach, okay? I think this is misspelled in your book, huh? No, that's right, methionine. Methionine, there we go. All right, so AUG, the start codon, it's not just a start codon. I mean, there's something there. It's methionine. You can still have AUG in other places on the mRNA. If it's a beginning, it's the start codon. If it's other places, it's just going to be regular methionine. But that is your indicator for start. Okay? So there are also 3 stop codons. Now, these codons don't code for any amino acids. It's like the period at the end of the sentence. This tells the transcription initiation complex to stop, detach, you are done, and go on about your way. So these are important in stopping the DNA sequence. What happens if you have a mutation where one of the codons gets changed to a stop codon? Like in the middle of your RNA sequence. It would do what to it? How would it mess it up?
Not code for the other half.
Yeah, it wouldn't code for the rest of it. So if you had a stop codon that got mutated into a stop codon in the middle of your DNA sequence, and you got problems because you're only going to make half your protein, and that protein is probably going to be jacked up and not do whatever it's supposed to do. So we're going to get into mutations later on, so I won't get too far into that right now. But it's important to have these codes and use these codes appropriately. On number 5, it talks about the use of inosine and the nucleotide that acts as a wild card when put in the 3rd position. That's because on most of these, like we said, even in that 3rd position, really doesn't matter what's there. It's still producing the same amino acid. So it helps, I guess, make it quicker. Y'all don't really need to know much about that. Part 6, universal for all organisms. Everything from viruses and bacteria all the way to humans and pigs and dolphins. And don't forget rats. All use the same amino acid chart. We're using the same stuff. Remember, viruses and people are viruses are basically hijacking our mechanisms to make more viruses, just like they do with every other living thing that they infect. So we're all using the same stuff. Same thing with bacteria, fungi, the whole 9 yards. Okay. Remember, RNA evolved first. It's unstable. It doesn't last very long. But it is important in the construction of proteins. Now, later on, we will get into viruses that have RNA at their core and some have DNA and what the difference is and how they work differently. All very important to understanding different diseases. Anyway, do we talk about COVID? Remember COVID, the 2 main vaccines that are out now are mRNA vaccines. Y'all know that? So the 3rd grade version of this would be they inject in you a mRNA segment and your cells make a piece of the outside of that protein that would normally be on the COVID virus. So your cells take in the mRNA, they make part of that protein that would be on the outside. Your body's like, hey, that's foreign, what the heck is that? And it starts to attack it and make antibodies and the whole immune response. And then it makes a bunch of memory cells. And then it's mRNA, so it's going to do what? Lumps of RNA after a little while. Yeah, it just breaks down. I mean, your cells will wind up breaking it down into different nucleotides and reusing it. And then it's done. It's not, contrary to popular belief by some folks, I don't think contains any strange nanobots that are going to be uploading data to your phones, but conspiracy theories run wild. All right, let's see here. He gets into unity and diversity, basically saying that since we all use the same amino acids, the same type of coding, the same mechanisms, that indicates that there is some connection between all life, which I guess we could agree on. Part A under number 6. Kind of the same thing. DNA is more evolved. It's more stable because of the double helix and the sugar. Reading frame. Remember, it's always read in the 5 to 3 direction. So mRNA is made in the nucleolus, just like DNA is replicated, and the other types of RNA that we'll get into, and then it's sent out to the ribosomes, either in the cytoplasm or the rough ER. So, you guys have a worksheet. Let me pass this out, and we're going to go ahead and work on this a little bit. We'll get started anyway. All 3 of them. You can put your name on here and keep this. Now, why do I have all of the DNA segment? Why do I have it partitioned off into groups of 3?
Because that's a codon.
Because that will make a codon, yes. So they're already partitioned off nice and neat for you. You people at home, I don't know if they are or not. I don't think so. Y'all can draw lines though. So here's what I want you to do. I want you to go through, and so this is your DNA strand. It's running in which direction? 3 to 5. So you're going to make an RNA that goes along with that. The RNA will be running which direction? 5 to 3. Okay? So make that mRNA strand that goes along with this.
You're still like right below it, or just?
Yeah, just right below it if you want to. I mean, you rewrite it if you want to, but I'd just put it right below it, probably the easiest.